There are many other classes of potassium channels that are expressed in neurons, and one of these is the two-pore potassium channel. This channel differs from all other types of potassium channels in that each subunit of the channel contains the P region, two distinct P regions. As you recall, for inwardly rectifying channels as well as voltage-gated potassium channels, each subunit had one P region, and thus 4 subunits were needed to make a complete channel. For the two-pore channels, each subunit contains 2 P regions, and thus only 2 subunits are needed to form a complete channel. These channels are open have a high open probability at the resting membrane potential of neurons. Therefore, they are termed leakage channels. They are responsible for the resting potassium conductance in neurons, and therefore they play an important role in generating the resting membrane potential of neurons, and we now believe that they contribute to the resting membrane potential of glial cells as well. They are voltage insensitive, and they are not blocked by normal potassium channel blockers. And like other classes of potassium channels, there are many subclasses of two-pore potassium channels, including TASC, TALK, TREC, and other types of two-pore channels. The TASC two-pore potassium channels are sensitive to external pH, and this is shown in the graph to the right, where as you increase pH, the open probability of the channel decreases. These TASC channels are also inhibited by transmitters, and this occurs via second messenger systems and the GQ pathway. And of course, with a decrease in the open probability of these channels, the TASC channels, this will result in a depolarization. The TREC channels function as mechanoreceptors in sensory receptor cells, and when these cells are stressed and the membrane is stretched, this leads to an open of the TREC channel. Other types of TREC channels are sensitive to temperature, and these function as the temperature sensors in the thermoreceptors. Inhalation anesthetics, including chloroform, halothane, ether, and so on, have a profound effect on both TREC and TASC channels, leading to an opening of the channels. And it's believed that inhalation anesthetics have their primary effect on neurons by modulating the open probability of these channels. So with an opening of these TREC and TASC potassium channels, the cell will hyperpolarize. This will decrease the excitability of the cells, lead to a block of action potential generation, and again is thought to be the mechanism by which these inhalation anesthetics work. Local anesthetics are also thought to operate by modulating two-pore channels, specifically TASC channels, but they do this in a different way. The local anesthetics are thought to close TASC channels. This leads to a depolarization and to depolarization block, which will also lead to a decrease in excitability of the cell. And local anesthetics may also block sodium channels directly in a use-dependent manner. That is, when the sodium channels are open during generation of an action potential, these local anesthetics can go into the channel pore and block the channel. We now turn our attention to voltage-gated calcium channels, which are found in moderate diversity in neurons. There are more different types of calcium channels that are expressed in neurons than sodium channels, but there are fewer types of calcium channels found in neurons than there are potassium channels. Calcium channels can be divided into high threshold and low threshold channels. The high threshold channels, or high voltage activated calcium channels, are activated and open at a relatively depolarized voltage. One has to depolarize neurons to a fair extent, to perhaps minus 20, 30, minus 30 or minus 20 millivolts in order to gate open these channels. About the same voltage that's needed to open up our classical sodium channels. There are several different types of high threshold calcium channels. These include the L-type channel, which inactivates, but inactivates relatively slowly over hundreds of milliseconds. And these channels generate calcium spikes in dendrites of various neurons. There's an N-type calcium channel, which inactivates somewhat faster. These N-type channels are found at the presynaptic terminal of neurons, as well as in other locations. There are also P-type channels, which inactivate slowly and are also found at presynaptic terminals, and they're found in the dendrites of Purkinje cells as well. Then if we look at low threshold channels, low voltage activated channels, there's one principal type, the T-type channel. This is a fast inactivating channel and inactivates at relatively with relatively small depolarizations as well. So these are the voltage-gated channels that are expressed on the plasma membrane of neuron, as well as other types of cells. L-type calcium channels are also expressed in striated muscle cells. However, there are other calcium channels that are found on the membranes of intracellular organelles in neurons and in other cells. Among these are the IP3 receptors, which are found on the endoplasmic reticulum of neurons and glial cells and are responsible for releasing calcium into the cytoplasm from the intracellular stores in the endoplasmic reticulum. Another important calcium channel is the ryanodine receptor, which is expressed on the sarcoplasmic reticulum in striated muscle cells. Calcium channels have many essential functions in neurons as well as in many other types of cells in the body, and as an exercise, which we'll be discussing in class, I'd like you to think of the various functions that calcium channels may have in neurons, glial cells, and muscles.